All right, finally, we have arrived at our last section of Chapter 2, the monster chapter for this semester. Proteins, nucleic acids, and the big wrap up. So, our last 5 learning objectives, we're going to talk about proteins and their importance. DNA and RNA, have a little bit of practice with different biological molecules that I have shown you and talked about. And then get to this question of monomers and polymers in the 3 major classes of biological molecules that have monomers and polymers. Remember, lipids don't truly have polymers because they don't get to be really, really big like we see in carbohydrates, proteins, and nucleic acids. Okay, so characteristics of proteins. Proteins are polymers of amino acids. So a polypeptide is a long chain of amino acids that are linked together by a bond called a peptide bond. And a polypeptide is sometimes used interchangeably with the word protein, but a protein is a biologically functional molecule that may consist of one or more of these polypeptide chains. So hopefully the next couple of slides will clear up a little bit of these words I'm introducing here. So let's start with the monomers of proteins. The monomers of proteins are called amino acids, and the reason they're called that is because they have an acid group, that carboxyl functional group, and they have an amine group, or an amino group. But not all of the amino acids are the same. They differ because they have a side chain called an R group. So this is your generalized structure of an amino acid. You can see over here on the left, we've got an amino group, that NH2. Over here on the right, we've got a carboxyl group, or an organic acid group. They're held together by another carbon atom. That's called the alpha carbon. The alpha carbon, because it's right there in the middle holding everything together. And then what makes amino acids different, all of this stuff is the same, but this side chain, or R group, is different from one amino acid to the next. And here they are, the 20 amino acids that we need to survive. No, you don't need to memorize all of these. Only if you're going to take a class in biochemistry would you really need to memorize all of these, or if that's what you want to do for your career. So why am I showing this to you? Here's what I want you to take away from this, is that this is how amino acids are different. You can see that part that's in sort of this light purple here, this lavenderish color. Those are all the same. So you've got that amine group, you've got that carboxyl group. The way that they're shown here, they have charges on them. Because remember, that acid group can donate a proton, and this amine group can pick up a proton. And that's the way that they normally are in the cell, is that they have these charges on them. But you should be able to recognize that there's, where'd it go? Here's an amine group. Here's a carboxyl group. There's the alpha carbon. And that's the same in all of these different 20 amino acids. So what makes them different is what's in the R group, or the side chain. And we group them based on the chemistry of the side chain. So in the case of these ones in yellow up here, they're all nonpolar, which means they're hydrophobic, which means you expect to see a lot of carbon and hydrogen. And that's exactly what you see. Here's just a hydrogen. Here's a CH3. Here's a bunch of C's and H's, more C's and H's. Here for a little bit of variety, there's an S. There's one little amine group in this ring here, but that's mostly just carbon and hydrogen. So all of those are considered to be hydrophobic. And then you've got these ones down here in green that have oxygen in them, and they're considered to be hydrophilic. They will interact well with water. So except for cysteine, has sulfur in it. And these guys actually have an amine group and a carbonyl group, so they're superpolar. Um, so that's going to affect how they behave in the big protein molecules. And then down here, we've got ones that have an additional carboxyl group on them or an additional amine group on them. And these ones, we call them electrically charged because when they are in the cell, they may have an additional negative charge on them and be acidic, or a positive charge and be basic. And you can see there's the amine group that, just like the amino group and the amino acid, is negatively charged. So another way to think of all of these amino acids being grouped together with their different side chains is, I'll show you that picture in a minute. But first, I just want to show you how do we put um, a peptide together, or a polypeptide, a group of amino acids, a chain of amino acids. They're put together by a different type of bond called a peptide bond. Now, you might remember we had a glycosidic linkage in carbohydrates when we made long chains of carbohydrates. We have another type of bond. It's the same process, dehydration synthesis, where we take that hydroxyl group that's part of the carboxyl group. We add that to a proton from the amine group, and bloop, here's a molecule of water. And when we take out that molecule of water, we form a bond between the carbon and the nitrogen, and that's called a peptide bond. So it's very important that all of these amino acids have a same basic structure, even if these side chains here are different. So another way to think about a protein is it's kind of like a train. Now, a train works by, well, yes, it has an engine, but then you also have a bunch of cars on the train. And it's very important that the cars have some commonalities between them. And here I've sort of analogized them. So you've got a carboxyl group on this car. It's attached to the amine group on the next car. And then it's got wheels and a chassis that's analogous to the alpha carbon. And then the different cars on the train are kind of like different R groups. So these are a couple of tanker cars, but in trains, they can have different types of beds on them. Like this one's got, a gravel open bed. These guys are like freight hoppers. You've got, I don't know what they're called, box cars. They're called box cars. And having all of these different types of tops to the train means the train can carry just about anything. It can carry liquids, it can carry solids, it can carry gases, it can carry lumber, it can carry all manner of things. But each type of thing that it might carry has a different structure to the top of the car, but it's absolutely essential that the wheels are all the same size and the connectors between the cars work together, or the train's not going to work. So a protein is kind of like that. So amino acids are linked together by these peptide bonds. If you have a chain of amino acids, it's called a polypeptide. And then a protein may be made up of one or several polypeptides. So it could be just a few amino acids, or it might be thousands of amino acids that make up a functioning protein. So here's where our train analogy kind of comes to an abrupt end, because not only do we need the amine group and the acid groups to fit together, but where those R groups come in is that they affect the structure when that chain gets all folded up and twisted and coiled, and it has a three-dimensional shape to it, which is something that you really don't want your train to do. You want your train to kind of stay in a straight line. So here's a couple of examples of how biochemists depict polypeptide chains. So they really do kind of look like this, where they'll have these twists and turns, and this looks like it might be a fun roller coaster to go on. But really what it is it's an example of what happens when the proteins get folded up into their functional shape. And in this model on the right here, which shows the same molecule, just with more of the atoms drawn in, you can see there's a lot of blue representing the nitrogen and the amine groups, and the red representing the oxygen and the carboxyl groups. But it's kind of harder to see all these coils and curves and grooves and everything on the ribbon model. But that's how polypeptides get their function, is once they're three-dimensional, which is something that, I mean, carbohydrates, there's a little bit of function in the helices and in the straight lines, but not like you see with proteins, which makes proteins able to do a lot more different kinds of functions in the cell. So the sequence of the amino acids is going to determine what that final three-dimensional structure is going to look like, and that structure is going to determine what it's actually capable of doing inside the cell. So, for example, one type of protein we have in our body are antibodies, which help protect us from disease. And the way that they do that is if we have, say, a flu virus, the flu virus has proteins on the outside of it. And our body has to learn to recognize those proteins from the flu virus, so our immune system can say, this is a bad guy, we need to destroy it. And how does it do that? Well, it has to make a protein that fits onto this protein from the flu virus. So you can see how our body is invented these new proteins, these antibodies that are kind of like a key in a lock. And this protein is like, is this protein a bad guy? And if it fits in there, it's going to trigger the body to destroy anything that looks like this flu virus. So the primary structure is just the sequence of the amino acids. Oh, another thing I meant to tell you about the amino acids is you may have noticed that they all have names. And those names get abbreviated. They can have a 3-letter name or a 1-letter name. And here you can see a series of the 3-letter names. Like here's glycine, proline, threonine, glycine again, threonine again, and so on and so forth. And all of these are representing different amino acids. There's 127 amino acids in here. So that's the primary structure. It's just like, what are the links in the chain? But then those peptide bonds in the chain can interact with each other. They can hydrogen bond to each other. So this is just in the backbone, just in the carboxyl and amino chain. You can get structures like these spirals. You can also, that are called alpha helices, or these pleated sheets that are called beta pleated sheets. And that's going to affect the secondary structure of the protein. So you have this spontaneous coiling up and sheeting action. But then for the tertiary structure, the third level of structure, what happens is the R groups, those side chains, interact with each other. So if you've got polar side chains, they may hydrogen bond to each other. If you've got non-polar side chains, they may cluster together because they're hydrophobic and they're like, ooh, get me away from this water. So the water will sort of drive them to be pointed at each other. You can have different amino acids with sulfur in them may bind to each other. You may get a covalent bond. That's called a disulfide bridge. Or you might even get an ionic bond between two different charged amino acids with their side chains. So this is the third level of structure, or the tertiary structure. And then, like I said, a functioning protein may be made up of several different amino acid chains. So here, for example, is hemoglobin, which we have in our blood right now, and it is carrying oxygen from our lungs through our circulatory system to all of our body's cells. So that's how our cells get fed the oxygen we need. What happens to that oxygen? That's a story for a few days from now. But a hemoglobin molecule is mostly made up of protein, which are all these squiggles and coils you see in here, which is the result of the secondary and tertiary interactions between the amino acids in the chain. But then hemoglobin is actually made up of 4 different subunits. So the final molecule has 4 subunits. Well, it's got 4 subunits, but it's 2 subunits that are repeated twice. So you've got 2 of these alpha subunits, alpha globin, and 2 of these beta subunits, which are in beta globin. And then proteins can also have other bits attached to them that aren't made up of protein. Kind of like, maybe you've got I'm trying to think. I'll think of something. But the heme group that you can see here at the top, that is not made out of protein, but it's like a protein, what we call a cofactor. And that heme group holds an iron atom in the center of it. Now, iron is not part of protein typically, but we can kind of add on these parts to make a functioning protein. And that's part of what we call the quaternary, or the 4th level of structure. So this functioning hemoglobin molecule relies upon having these chains of amino acids that are twisted up and coiled, but then we can also add on other fancy bits like heme groups and iron atoms. And it's actually the iron atom that binds to the oxygen to carry it throughout our blood. So quaternary structure results when you have 2 or more of these polypeptide chains to form one great big molecule. I didn't talk about collagen, but hemoglobin is a globular protein, or like a blob, consisting of 4 polypeptides, 2 alpha and 2 beta chains. Another thing that can happen with proteins is that if you change the chemical conditions, you can uncoil the protein. And sometimes you can reverse this, sometimes you can't. But when we change the shape of the protein, we call that denaturation. And when we denature a protein, you can see here it goes from being like just a big long squiggly ribbon, and then we do something like maybe we heat it up, or maybe we change the pH, or we add a bunch of salt, and all of that can change the shape of the protein. It changes the chemical environment of that protein, and it will change shape. This is something you've probably done and maybe not realized it. So if you ever hard boil an egg, the egg white is liquid. If you just crack an egg open like you're going to put it in your cake or your cookies or what have you, use it as a cooking ingredient, then it's liquid. It's a liquid protein. But then when you hard boil it, all of that ovalbumin that is the egg white denatures. And when it denatures, it forms a solid. And turns out that once you denature that protein that's in the egg white, and it affects the yolk too, you denature the proteins in the yolk as well, it doesn't go back, right? Have you ever tried to unhard boil an egg? Ever get in a situation where maybe you're doing some cooking and the only eggs you've got in the house are hard boiled, and it's like, well, I can't add this to my recipe because it's not going to work. That's because the proteins are denatured. But like I said, some proteins, you can denature them and then you can renature them and they'll sort of snap right back into shape. Okay, next video, I'm going to talk about how proteins are important to living organisms. And maybe you've guessed already that they're going to do some really cool things.